Hi, good morning, students. Welcome to our YouTube channel. Today, we are going to start another topic, that is physiology for first semester BSc nursing students in all universities in all over India. Okay, actually, all over India, all universities for BSc nursing, they are following the same syllabus only. Only the exam time and arrangement of topics will be different. Syllabus wise is similar only, so you can follow these classes. Even if you are studying in Kerala University, Rajiv Gandhi University, any university in India, because you all are studying the similar topics. Okay, already we finished the first unit of anatomy. If you didn't watch that videos, you can watch that 7 classes from day 1 to day 7. And once you visit, if you are not clear with the links, and directly go to playlist on our YouTube channel. There you can see first semester BSc nursing tuition, and then that you playlist you can watch one by one, day 1, day 2, day 3, like that till day 7 you can watch. And if you want classes in Malayalam, already I am taken classes in Malayalam also, Malayalam explanation also. If you want, you go to our YouTube channel and watch that videos also. And you want English classes means you can watch this playlist. Okay. Today we are going to start our first unit of physiology, that is general physiology. I think you all are familiar with the syllabus, the first unit syllabus. In that very important topic, one we are going to study today, that is cell cycle. Many exam repeatedly they are asking cell cycle for 5 mark and a 10 mark, part of a 10 mark question also. Okay, so you should study properly. It's very simple, but it's very important. Okay. Now about our channel, this channel is only for nursing students and nurses. So if you want, if you have any doubts, if you have any suggestions, any corrections, any messages, any comments, you can write in the message box. And if you like the video, please like it, press the like button. And if you didn't subscribe our channel till now, and if you feel this channel is useful, you can subscribe our channel, and please share with your friends also. Okay, today we are going to study cell cycle. Okay, let's start. You can see here, this is our syllabus. This is your syllabus, not my syllabus. Already I finished nursing. So this is our your syllabus, first unit of physiology. In that you can see the topics. First one is cell physiology include transportation across cell membrane. This is very important and repeatedly asked question. We will study in the next class. Transportation across cell membrane. Different transportation methods we will study on topic. Okay. And the second one is body fluid compartments, distribution of total body fluid, intracellular, extracellular fluid compartment, major electrolytes and maintenance of homeostasis. Actually, this is we are studying in anatomy and physiology as well as in fundamentals, but they are not asking questions in physiology. Usually, you will get questions in this from this topic in fundamentals of nursing in second semester. You will study fundamentals of nursing first in the first semester, but you don't have exam in fundamentals of nursing in first semester. The second semester only you will write the exam. Okay, so that time you will study this topic very detailedly. Okay, we are going for surfacely means no use only, so we will study this topic in fundamentals of nursing very detailedly. And the next topic is cell cycle. And last one important topics is membrane and glands. In all over, only 2 questions is very important. We can't avoid. You should study, and you may get questions in the public exam also. That is, there are transportation across cell membrane and the cell cycle. Already in the anatomy of the cell, we studied all cell structure of an cell and structure of organelles and the functions also I explained in that one. Here we will study transportation of cell membrane and cell cycle in this chapter. Then we will go to system based. First system, I think as per your syllabus, first system is respiratory system. First we will study the anatomy of respiratory system. Then next we will study physiology of respiratory. Like that, one by one we will study each and every chapter we will deal. Okay. I am following different textbook. I am getting notes from different texts, mainly P.R. Asha Lada and this one Rosan Wilson, and one more international books also I am using for the reference. So if you are watching these videos regularly, you will get all questions. I am promising you. You will get all the question for your public exam and your class exam. I will teach everything, each and everything of your syllabus topics. I will teach in this um, through this YouTube channel. Okay. Okay, so watch continuously. Don't skip the video. If you skip also, you will not understand the classes. That is the problem. Okay. Let's start our topic, that is cell cycle. What is meant by cell cycle? We studied about cell. Cell is structural and functional unit of our life. Structural and functional unit of life, that is cell. So what is meant by cell cycle? We will read some topics here. Many damaged, dead and worn out cells can be replaced by growth and division of other similar cells. Okay, in our body, in everyone's body, what is happening? Cell will be multiplying because of the why it has to be multiply, because each and every cell will die after some time, after their lifespan. It will die, some damage will occur, it will die and it will worn out. Okay, that damaged one or died one should be replaced with a new one. From where we will get a new one? From new cells we will get from the pre-existing cells. Because of the multiplication of the pre-existing cell only, we are getting new cells. With these new cells only, we are replacing this damaged or died cells. Okay, that process is known as cell cycle. Cell cycle we will study very detailed. This is one small introduction. Many damaged, dead and worn out cells can be replaced by growth and division of other similar cells. What will happen for our pre-existing cells? It will grow and it will divide into multiple cells. Then these new cells will replace damaged one. Okay, the frequency with which each cell division occurs varies with the different types of tissue. Different types of tissues will different lifespan. They have different lifespan. In sometime some cells will be dying more fastly, some cells will stay for long. Okay, so this division and cell division is not similar in each and every cell. Okay, this is normally carefully regulated to allow effective maintenance and repair of our body tissues. So, production and multiplication, growth of cell is everything should be controlled. It should be homeostasis. It should be equilibrium. Okay. At the end of their natural lifespan, aging cells are programmed to self-destruct and their components are removed by phagocytosis. This process is called apoptosis. Apoptosis is called as apoptosis. That means after normal life, each and everything will die, is it? While after aging, it will die. After normal lifespan, aging cells will self-destruct. What is it by self-destruct? Means destructing themselves. Okay, self-destruct themselves. It will destruct, and the damaged components or debris should be removed by the process of phagocytosis. Phagocytosis, and in the plus 1 plus 2 classes itself, you people may be studied. Okay, through the process of phagocytosis, it will be removed. Total in these process is known as apoptosis. Okay, clear? After aging process or natural life, after natural lifespan, what will happen to these cells? It will destruct. Okay, by himself or themselves, it will destruct. After destruction, this fragment should be removed by phagocytosis. This process is called apoptosis. Okay, cells with a nucleus have 46 chromosomes. In our body, each and every cell have genetic material. In that we have 46 chromosomes. That's one you people studied in the previous classes. Okay, what is our chromosome number? That is 46 chromosomes. And these chromosomes and cell divide by the process called mitosis. Maybe you people have the idea about mitosis and meiosis. Here our human cells are dividing by mitosis. Is a process that results into two new genetically identical daughter cells. Similar, this one cell divided into, this contain 46 chromosome, it divided into two cells. Each and every cell have 46. Similar daughter cells also have 46 chromosomes. Okay, the only exception to this is formation of gametes. Once our sex gametes, okay, like testis and ovum, so not testis, sperm and ovum, once sex cells forms during division, it will form haploid, means half number of half chromosome will be there, 23, 23, this ovum and sperm will have 23 chromosomes only. Okay, understood? Normally in our body, except sex cells, all other cells divide and form two cells, two identically daughter cells, and each daughter cells have 46 chromosomes. What will happen in the gametes or sex cells? Once it is divided into half, it will produce new cells which contain half number of chromosomes. That means 23 chromosomes. 23 chromosomes in ovum and 23 chromosomes in sperm. Okay, and this division takes place by the process of meiosis. Okay, this process is mitosis and this is meiosis. But usually you will get questions from mitosis only. What is meant by mitosis? What is the process of mitosis? How it is happening? What are the phases? Like that they will ask. The period between two cell divisions is known as cell cycle. What is meant by cell cycle? This our normal cell will divide it into half daughter cell. It will form new cells. This process is known as cell division. Between two cell division there is a gap. That space or that process is known as cell cycle. Period between two cell division is called as cell division. This cell cycle, this cell cycle has two phases. It has two phases. What are they? First one is mitosis and second one is interphase. Please note it, this one. Cell cycle have two phases. First one is mitosis, otherwise called M phase, and the second one is interphase. Okay, what you studied till now? Actually, we have our body is made up of many cells, million, trillions, many cells, structural and functional, functional and structural and functional unit of our body is cell. So each cell have their own lifespan. After lifespan, it will be damaging, it will be dying. So this aging cells will program to self-destruct. It will destruct themselves, and these products or fragments after the destruction, it will be removed by the process called phagocytosis. Okay, so this aged cells or damaged cell should be replaced with the new cells. Okay, from where we will get new cells? The pre-existing cell will divide and divide by mitosis or meiosis. Okay, mitosis or meiosis, it will divide and produce daughter cell. New cells will be produced. These new cells will replace our old cell or damaged cell. Okay, what is meant by where it is mitosis occur and where is this meiosis occur in our body? Except reproductive cells or sex cells or gametes, all other cells divide by the process of mitosis. What is the difference between mitosis and meiosis? Through the mitosis, it will produce two identical daughter cell and each cell have 46 chromosome. But in case of meiosis, in sex cells, what will happen? Once it divide, the daughter cells will contain only half of the chromosome. That means 23 chromosomes. Okay, so here we are going to study cell cycle. The space between or period between two cell division is called a cell cycle. This cell cycle have two phases. What are they? Mitosis and interphase. Mitosis and interphase. Mitosis otherwise called M phase and interphase. These are the two phases in cell cycle. This is the thing we have to study. This is a picture of cell cycle. Okay, this picture you have to draw while you are getting questions in the exam time. Okay, here you can see, if I draw, it will be making some issue. So I took this picture. This is from here we will start. Okay, this is mitosis, and this green phase is known as interphase. Okay, these are the two phases of cell cycle. This is the representation of cell cycle. In that two phases, mainly it is divided into two phases. One is mitosis, otherwise called as M phase or called as cell division, and the second one is interphase. Okay, here we will see what is meant by interphase, what is meant by mitosis, what and all is happening in each and every phases. Okay. Here you can see first phase is interphase. Okay, this is the longer phase. One word question. What is the longer phase of cell division, cell cycle? That is interphase. Interphase is the longer phase, and it has 3 separate stages. Okay, interphase itself, it have 3 phases. Okay. Next, we will see what are the phases in interphase. First gap phase or G1, it is called as G1, and second one is S phase. Third one is G2 or second gap phase. First gap phase, otherwise known as G1. Second one is synthesis of DNA or S phase, and the third phase is second gap phase. First gap phase, second gap phase, that is known as G2. These are the 3 phases of interphase. Next, we will go to picture and see. Here you can see this is the green one is interphase. In this first phase is first gap phase or G1 phase. This is the one. Second one is DNA replication or S phase. And the third one is second gap phase or G2 phase. Okay, these are the 3 phases in interphase. Okay. Then we will see what is happening in each and every phases. First gap or otherwise known as G1, the cell grows in size and volume. In the first gap phase or in the first G1 phase, what will happen to cell? Already I told you how the cell division occurs. The pre-existing cell will grow and divide. Okay. So that growth will occur in this G1 phase or first gap phase. The cell grows in size and volume wise and size wise. This cell will grow. This is usually the longest phase and most variable in length. Here you can see this is the most lengthiest phase, and this is the phase of cell growth. Cell growth, size wise and volume wise, cell will grow. Okay, cell will grow. It will not replicate. Cell will grow in the G1 phase. Understood? G1 phase means cell growth will occur. This is the longest phase in these 3 phases. Okay, the G1 is the longest phase. Sometimes cells do not continue round the cell cycle but enter a resting state. That is known as G0. During this time, cells carry out their specific functions like secretion, absorption. Okay, that one point is there. That one is an important point. You should know. Each cell, okay, each and every cell will undergo growth and growth will occur size wise and volume wise. But after this, usually these cells will go to S phase, but only some cells will go to G0 phase. That means resting state. That means it will not go for S phase. It will go to G0 phase. G0 means resting state. So it will not go for multiplication. Some cells will having absorption and secretion function. That cell will go to resting state. Okay, resting state, absorption and secretion. That type of special cells will go to G0. Not all cell, only some cell will go to G0. So you should mention, while you are explaining G1 phase, you should mention this G0 state also. Only some, very few cells will go to G0 state. That means resting state, and they may have absorption function and secretion function. Rest all cell after growth, it will go to S phase. Okay, next phase, that is S phase. Then next we will see what is meant by S phase. Here, synthesis of DNA will occur. Synthesis of DNA. What will happen? Synthesis of DNA. Synthesis means creation. The chromosomes, then cells have genetic material. Genetic material means it contains chromosomes. Here what will happen in the S phase? These chromosomes will replicate, forming 2 identical copies of DNA. Okay, already we have 46 chromosomes. Once this cell enter to S phase, again it will make one more copy of 46 chromosomes. So it is known as synthesis of DNA. We have to produce 2 cells each containing 46 chromosome. Is it? But actually we are having only 46 chromosome. One cell contain only 46 chromosomes. So in this S phase, it will form or create identical copies of DNA. Identical copies, one more DNA, one more set DNA, 46 more chromosome will create. Okay. Therefore, following the S phase, the cell now has 92 chromosomes. So after in the G1 cell had only 46 chromosome. On after entering S phase, during the S phase, it created one more 46 chromosome. Totally after S phase, how many DNAs or how many chromosomes are there in the DNA? That is 92 chromosomes. Enough DNA for 2 cells, nearly ready to divide by mitosis. Okay, now it has 92 chromosomes. Next, this will go here. You can from the G1 cell growth occurred. After G1, only few cells will go to G0 set. Rest of the cell will come to S phase. And the S phase, what will happen? Synthesis of 46 chromosome will add. Totally now 92 chromosomes. And next they will go to G2 phase. Next it will go to G2 phase. What is meant by G2 phase? Second gap phase. There is further growth and preparation for cell division. What will happen in the G2 phase? Further growth. Again, growth and preparation for cell division. Okay, now one cell came in G1, it grown, and it sometime it will go to G0 state, otherwise it will come to S phase. Once it came to S phase, it will create one more set of 46 chromosome. Now it is having 92 chromosome, and it will move to next phase, that is G2 phase. What will happen in the G2 phase? Again, this cell will grow and it will prepare for cell division. This one cell should be divided into 2 cells. Is it? For the preparation for cell division will occur in G2 phase. These are the 3 phases of interphase. Okay, clear? Once again, I will tell you. In the interphase is the longer phase, and the interphase itself is having 3 phases. First one is G1 or first gap phase. Second one is S phase, synthesis of DNA. And third one is G2 or second gap phase. What will happen in the first gap phase? In these 3 phases, G1 is the longest phase. Okay, in the G1 phase, cell will grow in size and volume wise, and this is the longest phase. And after G1, some cells will go to G0 phase for the resting phase. It is known as resting phase. That means some cells have the function of secretion and absorption. It will not divide. Okay, it will go to resting state. Okay, rest of other cells will go to next phase, that is S phase. S phase is known as synthesis of DNA. That means usually we will have 46 chromosome. In the S phase, again they will create or synthesis 46 chromosomes. So total 96 chromosome will be there after S phase. Next it will move to G2 phase. In the G2 phase, what will happen? Again, growth and preparation for cell division will occur in the G2 phase. Okay, this is all about interphase. Next we have to study what is the next phase? That is mitosis. Mitosis is very important. Mitosis. Okay, mitosis means the cell cycle, mainly 2 phases, interphase and mitosis. Interphase finish. Next we will study about mitosis, phases of mitosis. Okay, this is a continuous process involving 4 distinct stages. Again, this mitosis have 4 stages. Okay, what are they? Prophase, metaphase, anaphase and telophase. Okay, P-M-A-T. Okay, PMAT. You can study like this: prophase, metaphase, anaphase, telophase. Okay, prophase, metaphase, anaphase, telophase. These are the 4 phases in mitosis. First phase is prophase. Okay, first phase is prophase. During this stage, the replicated chromatin. What is chromatin? That is our chromosome. Replicated chromosome becomes tightly coiled. tightly coiled and easier to see under microscope. Each of the original 46 chromosomes or chromatid is paired with its copy in a double chromosomes unit. Two chromatids are joined each other at the centromere. The mitotic apparatus appears. This consists of two centrioles separated by mitotic spindle, which is formed from microtubules. The centrioles migrate one to each other of the cell, each end of the cell, and nuclear envelope disappears. And this is a main step, and this is a big step. Many things we have to study. What is meant by prophase? Here you can see this is prophase. This is the mitosis stages. Mitosis stages. This is prophase, metaphase, anaphase, telophase. Then last, these are the four stages, and the last one more process will happen, that is cytokinesis. Okay. Now we know after interphase, one cell have how many chromosomes? 92 chromosomes. Okay, these 92 chromosomes will condense or coil and form one thread-like structure. This is known as chromatid. Okay, chromatid, coiled structure. Then already we have 46 chromosome, again duplicate 46 chromosome formed, and each and every one by one from here on, and it will attach again, one attach like this. Two chromosome will attach together. Okay, it will attach to, this is the first one happening during this stage. Replicated chromatin or chromosome become tightly coiled, and original 46 chromosome is paired with the copy double chromosome. Okay, understood? Replicated chromosome and the original chromosome, one by one it will attach. One original chromosome and one duplicated chromosome, both will attach together. Okay, two chromatids are joined with each other at the centromere. Centromere means, you know, this is chromosome. This center part is known as centromere. Okay, like this it will attach. It will attach by a centromere. Okay, to one original chromosome and one duplicate chromosome, means created one double chromosome will, and it will attach. Two chromosomes will attach and form a double chromosome unit. It will attach like this and form a double chromosome unit. Where it will attach? It will join at the each other at the center of centromere. While attached to it will attach by centromere. Two chromatids will join by centromere. Okay. Two centrioles after offer. So now how many chromatids are there? Means bonded one will be 46 original and 46 new one. Each and two chromosome will attach, and again 46 units will be there. Double chromosome unit, 46 double chromosome unit will be there in the cell. Understood? One by one, one original, one duplicate will attach and form one double chromosome unit. Like that, how many double chromosome unit is there? 46 double chromosome unit will be there. Okay, and next what will happen in our cell? Each and every cell we studied, we have centrioles. It is one special structure, that is centrioles. You can see this is centrioles. These two centrioles will go to each end. Okay, next it will go to one upper pole and it will go to one pole. Okay, during cell division, this centriole this centriole have the role only during cell division in our cell. Centriole will go to each pole. One will go to this pole and one will go to this pole. Otherwise, one will go to this pole and one will go to this pole. Like this, it will go to two poles, and these centrioles will be attached with a mitotic spindle. Okay, mitotic spindle. It will be attached with a mitotic spindle. Here what happened? Original chromosome and a newly created chromosome, one by one it attached and it formed a double chromosome unit. Like that we have 46 double chromosome unit, okay, in the cell, in the genetic material. And the cytoplasm, we have centriole. This centriole will move to each pole or each end of the cell, and these centrioles will attach by that thread-like structure, that is mitotic spindle. Mitotic spindle. Okay, this and all happening in the prophase. This much is happening in the prophase. You understood what is happening in the prophase? Already 92 chromosomes is there. It will form into 46 double chromosome unit, and this centriole will go to each end of the cell, and this centriole will be attached by mitotic spindle. This all happening in the prophase. Next we will see what is happening in the metaphase. Okay, metaphase. And one more thing it will happen, that nuclear envelope will be disappearing. Here you can see this is nuclear envelope, nuclear membrane, outer covering of our genetic material. This nuclear membrane will disappear in the end of the prophase. This nuclear envelope will be disappearing. Okay, nuclear envelope will be disappearing. Okay, this and all happening in the prophase. Next, we will see what is happening in the metaphase. Metaphase. In the metaphase, this chromatid align on the center of the spindle attached by the centromere. Okay, what will happen? This envelope is disappeared. This all chromatids will come in the center of the, this is centriole and this is mitotic spindle. In the mitotic spindle, center of the mitotic spindle, these all chromosomes will arrange. Okay, like this it will arrange. Okay, these chromatids will arrange in the center of the mitotic spindle. In mitotic spindle, it attached by two centrioles. These centrioles are present in the polar region of cells. Okay, like this, one end and another end, both end it will be centrioles will be located in the both end of the cell, and these centrioles will be attached by spindle, and these chromatids will be arranged in the center of the spindle. This is happening in metaphase. Okay, what is happening in the metaphase? Already in the prophase, nuclear envelope disappeared. So after placement of these centrioles in the both end of the cell, these centrioles are attached with a spindle, mitotic spindle. These chromatids or our chromosomes or double chromosome unit will come and attach or place in the center of the mitotic spindle, which is connected the centrioles. Okay, centrally it is aligned or arranged. Okay, this is happening in the metaphase. Next we will see what is happening in the anaphase. What will happen in the anaphase? Anaphase, see here you can see the centromeres separate. One of the each pair of sister chromatids migrates to each end of the spindle as the microtubules that form mitotic spindle contract. What is happening in the anaphase? Here you can, the picture you can see, it will, this is centromere, this is centromere. In the centromere, it will divide like this, it will divide through the centromere, it will divide into two equal halves. Understood? Two equal, it will be, these chromatids will be dividing into two equal half because of the division of centromere. It will become half like this. Okay, now this spindle are separated and this spindle will contract and go to this centromere. What is happening here? You can see after separation from here, this half chromatids is going to each centromere. Understood? It will divide here. It's divided here and it will go to each centromere side like this. It will go. Okay, this is happening in the anaphase. Okay. Next what we will next phase is telophase. Then next we will see the telophase. What is happening? Mitotic spindle will be disappear and chromosome will uncoil and nuclear envelope will reform. Okay, once again we will see here. This spindle was there, no, mitotic spindle, it will be disappearing and the coiled chromatids will uncoil, and this nuclear envelope will form. It will start to form a nuclear envelope. Okay, nuclear envelope means outer covering of nucleus. Nuclear envelope will form in the both side. Okay, mitotic spindle disappeared. This chromatids will uncoil and form chromosome, and it will form nuclear membrane or nuclear envelope. This is happening in the telophase. And cytokinesis, next phase is cytokinesis. And some books it's telling like it is included in the telophase only. In the some book it is telling us cytokinesis other process. In the both you can write, no problem. Telophases, you can add cytokinesis in the telophase. Otherwise you can give another heading as cytokinesis. That also you can, it's acceptable only, no problem. Okay, what is happening in the telophase? Mitotic spindle will disappear, chromosome will uncoil, because already in the first phase prophase is coiled, and it will uncoil, and there will be formation of nuclear envelope or nuclear membrane. And the next step is cytokinesis. What is meant by cytokinesis? Now everything happening in one cytoplasm. So to divide these cells, the cytoplasm also should be divided. This division process is known as cytokinesis. So this cell membrane and the cytoplasm will divide. Now two nucleus formed, and this cytoplasm should divide into two. This division is known as cytokinesis. Okay, here you can see cytokinesis occur. Cytosol, that means our cytoplasm, intracellular organelles, cell organelles, plasma membrane, everything will divide into two and form two identical daughter cells. This last process of cytokinesis, it will divide. Cytosol will divide into two, plasma membrane will divide into two, and it will form two identical cells. Two identical cells. This is all about cell cycle. Okay. Once again, when I will tell you the mitosis process, interphase, you know, you are clear. G1 phase, growth will occur. S phase, additional 46 chromosomes will form, and it will make 9. Now it is 92 chromosome. In the second gap phase or G2 phase, again growth and preparation for cell division will occur. This is all about interphase. Next, mitosis, 4 phases: prophase, metaphase, anaphase, telophase. In the prophase, what will happen? This chromosome, one original chromosome and one newly formed chromosome will attach, and it will form a double chromosome unit. Like that, 46 double chromosomes unit will form. And these centrioles will go to each end of the cell, and it will be attached by the spindle, and this nucleus membrane will be disappearing. Okay, after disappear, in the metaphase, these chromosomes, our coiled chromosomes or chromatids, will arrange in the center of spindle in metaphase. In the anaphase, in the third phase, it will divide through the centromere. It will divide into two equal half. These chromosomes will go to what? After a division, spindle will contract and it will go to centriole side. It means each end of the cell. So it will divide and go two sides. And after division of chromosomes, this chromosomes will uncoil and nuclear membrane will be starting to form. Okay, after nuclear membrane formation, this cytosol or cytoplasm and the plasma membrane will divide and to form through the cytokinesis process, and it will form two identical cells. Each cells have 46 chromosomes. This is mitosis process. Okay, this is all about cell cycle. It's very, very important. You draw. So if you are getting cell cycle question, put a introduction like this and draw this picture, and you can tell like two phases like interphase and mitosis. Then you can start with interphase, 3 phases, just explain. Next you can start with the mitosis. When you are writing mitosis, each and every phase you draw the picture also. This picture also you can add one by one. You can, in the prophase you can add this picture, metaphase this picture. No need to draw all picture together one by one. While you are explaining one phase, you can draw one picture, and the next phase next picture. Like that you can put, while you are putting cytokinesis, you can draw this picture and write. It will form two identical cells. Okay, If you like the video, please like the press like button, and if please share this video with your friends also, and study well. Actually, you will not get a, you don't wait for classes, study as much as you can. Anyway, you people only going to study in the end of or during university exam, so you people will not, only 6 months is there, 6 months will go like anything, it will go fast. Okay, you will, it is not enough to study 4 subjects, so many things are there, but you, if you are ready and if you are, if you have the interest, it's more than enough. Okay, so you can study very well, very easy topics if you are interested. Okay, I will explain you, I will give each and every topic classes for you through YouTube channel. Only thing, you should not skip the video and watch continuously, and if you have any comments and suggestions, please mention in the message boxes. Okay, and share with your friends also. Thank you for watching. Bye bye.